Hello, nice to see you today. My name is Paul. I'm a physician at the Necker Hospital working in the pediatric immunology department, and I also work at the Institute Imagine in the laboratory of human genetics of infectious diseases. And today I'll talk to you about an old concept that we're trying to reestablish as a new one, which is autoimmunity against immunity. So I have no conflict of interest, and the global idea of our research is to understand the immune predisposition to infections. So overall, in the field of infectious diseases, there is a sort of U-curve in the prevalence of severe disease, with more severe cases in patients that are younger, or at the other extreme, which are older or even very old. So nevertheless, this being said, that doesn't explain why some elderly patients have more severe infections while others don't. So in the context of COVID-19, we wondered why patients died of COVID-19. And for this, we recruited patients either in the ICU, like you can see here, in an ICU in England in 2020, to sequence their exome or genomes, or to compare patients with asymptomatic or mild infection. And in a very biased manner, we looked for genetic defects in a pathway which is a type 1 interferon pathway. So type 1 interferons have been named interferons because they can interfere with viral replication, and this is why Isaacs and Lindemann named them so in the 1950s. What's quite surprising is that there are 13 different subtypes of interferon alphas and 4 other circulating interferons, and it's still a mystery why there are so many different subtypes while they all bind to the same receptors, interferon alpha receptor 1 or 2. And what is known about this type 1 interferon pathway is that genetic defects in the pathway leads to severe infection, including severe influenza and pneumonia. So we wondered if inborn errors affecting the type 1 interferon pathway could lead to severe COVID pneumonia. What was very surprising is that we indeed found that about 2 to 3% of patients in the ICU because of life-threatening COVID-19 had a genetic defect, either partial or complete, in the production or response genes of the type 1 interferon pathway. What was even more surprising is that we found a few adult patients with complete deficiencies, as you can see here, and the cells of these patients do not respond at all when we stimulate them with interferon alpha 2 or interferon beta. In children, we find that this is even more prevalent, and we find that more than 10% of children have a genetic defect affecting the type 1 interferon pathway, and that it's almost always a complete defect. But this remains a still number and only was found in patients below 65 years of age. So next, we wondered if other forms of deficiencies in the type 1 interferon could lead to severe COVID, and for that we turned to their autoimmune phenocopies. We looked at autoantibodies against type 1 interferons. And here, what is very surprising is that colleagues from the 1980s were already discussing about these in patients with interferon alpha 2 treatment in the context of hepatitis, or interferon beta treatment for multiple sclerosis. They're also found in a small percentage of women with lupus, and we'll discuss this later, and in patients with rare diseases such as thymoma, or extremely rare genetic diseases affecting the AIR gene. Overall, everyone thought that these autoantibodies were clinically silent, yet there was one paper in the 1980s which found autoantibodies blocking type 1 interferons in a patient with severe VZV disease. So knowing the known role of type 1 interferons to fend off viruses, and the previous finding of a patient with a severe disease, in addition to having genetic defects in patients with severe COVID, we looked for these autoantibodies neutralizing type 1 interferons in patients with severe COVID. And here in 2020 and then onwards, we surprisingly found that more than 10% of patients in the ICU because of COVID had high titers of autoantibodies neutralizing interferon alpha 2 and or interferon omega. By contrast, patients that were asymptomatically infected did not have antibodies, or these were extremely rare. And then we quickly looked in the general population in healthy controls, and found that in this population below 65 years old, the autoantibodies were quite rare, between 0.2 and 0.3%. What was interesting also is that most of these patients were men and most were over 65 years of age. Yet, we even find them in children with COVID pneumonia in more than 10% of cases. And finally, people were thinking that these autoantibodies could have been triggered by infection, and so we tested all the samples we could find from before SARS-CoV-2 infection in patients with autoantibodies, and found that in all the cases, the autoantibodies were already present before SARS-CoV-2 infection. Then we and other colleagues replicated the study worldwide, and there does not seem to be any geographical or ethnical bias that explains the occurrence of these autoantibodies. But really, to show that these autoantibodies were the cause of severe infection and not just associated with it, we teamed up with Charlie Rice's lab at Rockefeller, and they performed a very elegant experiment where they infected control cells with SARS-CoV-2 in presence of interferon alpha-2 and plasma, either of control, the two blue lines. And here you can see that the interferon alpha-2 completely blocks SARS-CoV-2 replication. By contrast, plasma of patients with autoantibodies block the protective effect of interferon alpha-2, leading to an extremely high viral growth. And what they did was to perform serial dilutions, and you can see here, even when you dilute the plasma 100, 1,000, 10,000 times, you still see this neutralizing effect against interferon alpha-2. So these autoantibodies lead to higher viral growth and have an extremely high avidity and affinity, and we're now proving with colleagues here at Necker that the B cells producing the autoantibodies indeed pre-existed COVID-19 infection. And so next, knowing that these autoantibodies pre-exist SARS-CoV-2, we wondered what is their prevalence in the general population. And so here it's one slide, but it's actually, it was a lot of work. We tested 34,000 individuals of all ages from different cohorts in France and abroad, and found that both in children and young adults, the prevalence of the autoantibodies is relatively rare, 0.2 to 0.3%, but this increases strikingly with age, reaching more than 4% between 80 and 85 years old. Then there's a striking decrease of these autoantibodies. We don't have the proof yet, but it's perhaps because when the autoantibodies arise with age, the patients unfortunately die of infection because of these autoantibodies. In any case, the risk of having a severe COVID-19 pneumonia when you have these autoantibodies is extremely high. Now, we next wondered, what about other viral diseases? So we found that these autoantibodies were responsible for 1/3 of adverse events following yellow fever live attenuated vaccine, and about 5% of patients with severe influenza disease. And more recently, we were interested in the rare cause of severe encephalitis by West Nile virus. What's interesting about West Nile is that it's a flavivirus like yellow fever, and it's actually quite endemic in many countries, especially in Eastern Europe and Southern Europe, and can give either very mild symptoms or, on the other end of the spectrum, very severe encephalitis with 5 to 20% of mortality. What's interesting epidemiologically is that while the seroprevalence is stable overall, the clinical incidence is actually rising with age, like the one of autoantibodies against type 1 interferons. So here again, we hypothesized that autoantibodies neutralizing type 1 interferons could lead to severe encephalitis, and we tested a cohort of about 600 infected individuals, either asymptomatic, the black dots, or with very severe viral disease, the red ones. Here the results are shown at the opposite as last time. It's we look at the activity induced by type 1 interferons, and so if you're in the upper part of the scheme, it means that the activity is high and there's no neutralizing autoantibodies. So no patients with a mild form had neutralizing autoantibodies against type 1 interferons, while 40%, which was extremely striking, of patients with an encephalitic form had autoantibodies blocking interferon alpha-2 and or interferon omega and rarely interferon beta. It's a neurological disease, so we had access to CSF sample, cerebrospinal fluid samples of some patients, and we found an extremely good correlation, meaning that in patients with autoantibodies in blood, we always found them in the CSF of these patients, and we made small calculations, and we think that the autoantibodies could even be at a higher concentration in the CSF than in blood. Now, obviously, these autoantibodies lead to an increased infectious risk because of viral diseases, but could they have any beneficial effect? And so a team in the internal medicine here in Paris looked at these autoantibodies in a cohort of women and men with lupus, and very interestingly found that the patients that were harboring these autoantibodies in red here had a lower disease activity with 0 flares during the time of the study, almost 3 years, while the patients without these autoantibodies had a higher disease activity and more flares than the patients with autoantibodies. And interestingly, now there's a new treatment approved, which is anifrolumab, which is an anti-IFNAR1 antibody, and this is mimicking basically the effect of these autoantibodies to block circulating interferons. The contrast, the downside of this is that these patients with autoantibodies here in red have a higher rate of zoster infection that can be actually quite severe, and also of severe COVID-19 infection as described previously. So now, if we look at the potential effect of autoantibodies in humans, I've discussed here neutralizing autoantibodies, but the effects can be much larger. It can activate receptors, deplete specific cells, lead to inflammation, and have an anti-tumor effect, or clearing certain molecules. And so today in the lab, we're trying to introduce or reintroduce this concept of autoimmunity against immunity, which made us think of an Ouroboros, which is a Greek, from the Greek mythology, this snake that eats its own tail. So what do we know about autoimmunity against immunity in terms of autoantibodies? Well, the most common one, the one I see in the clinic almost every day, are autoantibodies to neutrophils. Autoantibodies neutralizing, leading to the opsonization of neutrophils. As you can see here, a neutrophil being destroyed by a macrophage, and this happens in young children and usually leads to relatively benign staph infections. What is much more severe are autoantibodies against the complement system. So in the 1980s was described autoantibodies against C3nef that would actually stabilize which are actually stabilizing, leading to an activation of the C3 convertase and leading to depletion in C3. And so these patients unfortunately had very severe infections because of meningococcal meningitis or streptococcal pneumonia. And these had the equivalent of a genetic defect in complement, but because of an autoantibody. Now, if we go back to cytokines, we in pediatric immunology, we see patients with severe mycobacterial infection who have genetic defects in the interferon gamma pathway. And in adults, colleagues in the US described a new form of what they called adult immunodeficiency in patients usually from Southeast Asia because they have a very specific HLA haplotype that suffered from very severe either mycobacterial or other intracellular infections. What they found, which was very surprising, is that most of these patients had high titers of autoantibodies neutralizing interferon gamma, as you can see here. And these patients do not respond fully to antimicrobacterial therapy, and so they need to be treated by a B-cell depleting agent, which is, I guess, kind of counterintuitive when you think that you're treating an infectious disease. So if we summarize the field of autoimmunity against immunity today, there's the autoantibodies against neutrophil, autoantibodies against complements, and then if we look at the autoantibodies against cytokines, I talked about type 1 interferons leading to severe viral infections, autoantibodies against interferon gamma, type 2 interferon, leading to mycobacterial infection, for example. There's autoantibodies against IL-17, leading to candida infection or other fungi. autoantibodies against GM-CSF, leading not only to pulmonary alveolar proteinosis, but also to crypto infection or nocardia infection. And finally, autoantibodies against IL-6, leading to very severe bacterial infection with a very specific phenotype because these patients have no inflammatory syndrome with a negative CRP, as patients who would be treated with an anti-IL-6 receptor antibody. So this was in 2024, and during the year of 2024, 2 new autoantibodies against cytokines were described. In adult patients with other intracellular infections, autoantibodies against IL-23 were found. And in pediatric patients with a pediatric form of inflammatory bowel disease resembling Crohn's disease, our colleagues in England found autoantibodies neutralizing IL-10. And this was very surprising because these patients did not respond to conventional therapies of Crohn's disease, but responded fully to rituximab treatment. So it's really a shift in our way of thinking. So to summarize, the concept of immunity, autoimmunity, which we like to call Ouroboros, it's a little bit counterintuitive because it's adaptive immunity attacking intrinsic immunity, but potentially for COVID-19, it could explain 2 million deaths worldwide because we found these autoantibodies in 20% of people who died of COVID-19. They're found mostly in the elderly, which could explain part of the risk, but also in younger individuals and including in children. Obviously, we'd love to find these autoantibodies as early as possible to prevent the infection because these patients respond well to mRNA vaccines, for example, and also to avoid giving them live attenuated vaccines. But in case they get infected, well, you can use any therapy that you have at disposal to treat the infection. And in a few very severe cases, we've even tried removing these autoantibodies, which is an option to be discussed. Now, the big questions that are open is what other viral and non-viral infections or non-infectious phenotypes can we explain because of autoantibodies against immunity? Who should we screen and when? Should we screen all the elderly individuals for autoantibodies against cytokine interferons? This is really a question for the public health authorities. What treatment can we give these patients? Can we block these autoantibodies specifically, or can we remove the B cells producing them? That, I guess, is for the future. And we're working on the genetic causes of these autoantibodies, and we think that the common cause is perhaps because of a thymic defect, either a genetic, germline, or somatic. Now, I'd like to thank you very much, merci beaucoup, to the FAI²R, all the physicians and the patients who participated in our studies, because without you, none of this is possible. The whole lab that's headed by Jean-Laurent Casanova and Laurent Abel, and all my colleagues here in blue and in green with whom we've done these studies and we're still continuing, my clinical colleagues. And with that, I'd like to thank you very much, and I'd be happy to discuss further, either to study patients or to exchange new ideas. Thank you.